So I will talk about things more relevant for the future. So not about the old stuff, so more about the future. And you know, the future means that in sooner or later, these quite interesting things in the gene and cell therapy, which are currently more in the academic field, will enter now the area of manufacturing. And this is what my talk will go about. The qualification, the extractable leachables qualification, in case you start to use a gene and cell therapy product, or you want to produce a gene and cell therapy product. It starts a little bit with highlighting the special challenges which are there. Then I come to the question whether extractables data are suitable, whether extractables data what we are having today are suitable. So that means, for example, the USB 665 data. Then I come to my favorite topic. This is this exposure estimation of extractables, and now, of course, in the gene and cell therapy area. And then I will show you some very interesting work we did together with the Max Planck Institute to assess a little bit the question how critical are extractables in this particular gene and cell therapy application area. And then I come to a conclusion. Let's start with the challenges. So in gene and cell therapy, we are commonly, it's quite a little bit different to the common biopharmaceutical processes. So we take cells from a donor, for example, then we do something with the cells, then during that time these cells are ex vivo. They are in close contact with plastics, of course, because we do that with pipettes and with dishes and with bags and so on, bioreactors, you name it. And then finally we transfer these cells back into the patient. As I said, we are heavily relying on single-use devices here. Nearly everything is made in single use in this gene and cell therapy area. We know this is what we are talking about here. Of course, plastics may be sources of process equipment-related leachables, and compared to a classical biotech application, there is not so much purification along the process. We have some washing steps, we have some media exchange and things like that, but we do not have this really very efficient downstream processing what we normally apply in common biopharmaceutical processes, in most of them. When you think about gene and cell therapy, then it becomes immediately clear you must consider more than in a biopharmaceutical process. Patient safety risk, okay, you have to consider that, this is clear. But now process performance becomes key. Because, as I said, you are manipulating cells ex vivo. So how well is this process operating? And of course, you have to consider the quality attributes of these cells. So, so to say, a part of the patient is outside the patient, and we treat them, and it comes in contact with plastic. So this is where our thinking must go to when we want to assess a gene and cell therapy application. Not only patient safety, process performance and quality attributes are now key. I think it's not necessary to explain that too much in that audience here, with that audience here. What do authorities commonly want us to do? We saw the Annex 1 before. Authorities want us that we have extractables data, qualitative and quantitative extractables data, ideally without unknowns, of course. Then authorities want us to transfer this extractables data to exposure data. And in this case, it means exposure to the cells, exposure to the product, which is again the cells, and to the patient. And then we shall compare our calculated exposure with thresholds and limits. In case of cells, these are EC50 values. In case of patients, these are PDE values or TTC values. Calculate a safety margin, and then we can say whether our process is safe or not, or we have to do something else. And now in gene and cell therapy area, first question, are the extractables data suitable what we are having today, or do we need new data, other standards? The next question is, are our methods which are used for exposure estimation sufficient for this field, gene and cell therapy application? And last but not least, a tricky one, are our toxicological data and biocompatibility data we are having, are they sufficient for gene and cell therapy application? It's quite questionable, I think. But let's start with extractables data. And we heard a lot about USP 665. I personally would say the gene and cell therapy area fits very well into that USP framework here. We will test single-use components. This 40 degrees is, of course, approximately that what also occur in gene and cell therapy applications. We have contact times of 7 days, 21 days. This fits also very well to USP 665. We at Sartorius have, in addition, pure ethanol extracts. This is quite good for us for the qualitative analysis of potential extractables. And then you have the surface to volume ratio in USP 665. So from my point of view, data from an USP 665 test with the appropriate analytics behind is able to fulfill the requirements of the gene and cell therapy application. So we can make a nice cross there. In particular, if we consider our history. So we came in the 2010s with extractables profiles which were only partially elucidated. Today we have extractable profiles. So here I have the year 2020 extractable profiles which are 100% elucidated. So we know all the compounds which may occur from a bag and tube and filter or whatever. So there are no unknowns anymore in extractable profiles. This is something we need to realize, and this is a good thing to know, because this allows us now extrapolations and exposure estimations. Now with a physical chemical understanding, you can now transfer the data to other conditions, and in particular our toxicologists can use PDE values and TDE values for the assessment, which is of course of much more use than to use TTC values or something like that. But again, you need fully elucidated extractable profiles. But as I said, this is almost achieved. And the other thing is what we at Sartorius are doing, we combine all our extractables data in a database. So we have our extractables data together with physical chemical information on the extractables data, together with tox information. By the way, we have today 180 PDE values for the typical extractables, so this covers almost all extractables already. So it's not necessary to use TTC values anymore. And this allows us then to produce standard extractables guides, so applying exactly the extraction conditions, and then you get extractable guides. But this allows us also with the PhysChem data to scale extractables information and to combine, aggregate the data for assemblies. This answers a little bit the question from Simone. Also, yes, with physical methods, with algorithm, you can of course combine devices, components into an assembly and produce an extractables profile for an assembly which fits then very well. And this system allows us also, because we have a lot of data inside, we can make a nice mathematics with the data. We can extrapolate to various in-use conditions regarding the extraction strength of the media and the size of the device. And this is, I will come to that very soon. I would say we can answer the first question. Yes, the extractables data we are having today are fit for purpose, so we can use them. In particular, if you have them aggregated them in a kind of database and you can make use of prior knowledge, so to say. And what I would really stress out here is we do not need further data like DMSO data or data on low temperature. Now, this is definitely useless. DMSO water mixtures are a very weak solvent. If we have data from ethanol, like our data, and data from ethanol 50-50, this covers this DMSO. And what a lot of people not really understand is DMSO is a concentrated DMSO is a very critical solvent because it discriminates your extractable profile. It prefers to extract aromatics over aliphatics, just to make that clear. And with ethanol, it's different. Ethanol extracts any compound until a certain partition coefficient. Okay, so that means this area, I think we can answer with a clear yes, extractables data are fit for purpose. Now, the question, are the exposure estimation methods are fit for purpose? I've shown you that plot already. I said, okay, with our extractable simulator, we can calculate this grid surface, which gives you the exposure surface in a static process, adjusted to the size of the single-use device and adjusted to the liquid composition and the temperature. And the green surface here is a kind of threshold. This is just for an example, a threshold for 2,4-diterts butylphenol in an application with 200 milliliters. Then you see they have the green surface, and all the data and all the applications which are below this green surface area is an application where you are operating in the safe area. So this can be calculated. But as nice as it is, this is not sufficient if you talk about gene and cell therapy applications. Why? Because this covers not dynamic processes. And I said in the beginning, a gene and cell therapy process uses media exchange, uses some washing steps, and we have to consider that now. And the other thing is, of course, such a model can give you no indication on the exposure on cells. In the beginning, I highlighted this exposure question, but we cannot really, this is only information about the liquid phase. A liquid phase without any biologics in. So we need some information about the cell, so that means the model must be extended. Now let's extend the model, and here I have an example for an perfusion bioreactor as it is used in allogenic therapies. This bioreactor in the exposure assessment must now allow to show how the migration out of the pearls of the contact materials happens, how the flow through the system is influencing the concentration. It must also include the pearl distribution between the liquid phase and the cells, of course, because the cells is now our product. Yeah? And it must consider the growing biomass. These are the parameters which must be considered in a reasonable risk assessment here. What I would highlight in addition is a traditional leachables assessment using analytical methods cannot treat that system appropriately because the measurement is not possible. If you ever have tried to measure leachables on T cells, for example, then, as I said, you need a PhD student doing that. You can never do that on a regular basis. This is really a hard job. So this is the problem where we are in it, and in the yellow box, these are the things we must solve. And we can solve that by developing a kind of digital twin of this allogenic bioreactor. Here we need to consider that the pearls are in this system, in the system box. This is, of course, the bioreactor, the gray box with the red devils. This is the single-use device with the pearls inside. Then these pearls may migrate out of the system. So this is something we need to consider. We need to consider that these compounds are then dissolved in the liquid phase. They may be distributed between the liquid phase and the cells while the number of cells is growing. So this is the assessment we need to make if we want to get reasonable exposure estimations for extractable leachables in a quite simple allogenic bioreactor. And in addition, we have to consider that there's a flow through the system. Of course, it's not that big deal to find some physical and mathematical equations they can describe migration, flow, adsorption, and growing biomass. This is just examples, for example, for the growing biomass. I've just assumed the Van Hulst equation. You can, of course, use other equations for that. You combine it in a model equation, then you get a digital twin out of that. And for the digital twin, you need then some input data, and some input data can be used from our extractable simulator, so all the information on extractables and the geometric information from the extractable simulator. You need the process information. This is, of course, known. You know how long you will operate your bioreactor. This is clear. You know the temperature, you know the flow in the system. Then you need some information about the cell growth. Here you have the different ways to get information. You can either use real-time data. This is a possible way to go. You can use something like this Verhulst equation I've shown before, or you can even take them can model them with certain models which are also available today. So to be honest, we are in the year 2024, so we are already in a situation that for various processes there are digital twins available. So for a bioreactor, you can get a digital twin which can calculate the cell growth and things like that, just to have that said. And last but not least, you need a tricky number. This is the adsorption coefficient of the leachable with the cells. And here you have only one possibility. You have to make a dedicated experiment. And I had a master's student, a PhD student who did some work on that for me, for T cells and CHO cells and some other cells. This is published. And there also a nice protocol is published there, giving you some guidance how you can determine these adsorption coefficients. And then you have a digital twin, and this digital twin can then calculate the trajectories of the pearls along the process while you are expanding your cells. And for any time point, you can nicely see what is the concentration of the pearls in the liquid phase, what is the concentration of the pearls adsorbed on the cells. This is a number where we are interested in. And I have given here also the growing biomass, as I said, a simple sigmoid curve here. This is then what you can see here. What you can also see here is that such a system, such a perfused system, is washing out the pearls, obviously. So you see that the pearls are going down over time in the liquid phase as well as in the adsorbed on the cellular phase. So perfused systems, this is also a learning at this point here, perfused systems cannot accumulate pearls because the release rates of pearls from plastics is always orders of magnitude lower than the perfusion rate. Okay, so I would say we can also answer this question with a yes, but of course we need to maybe we need to leave a little bit our comfort zone with check boxes and so on in the assessment. Now the assessment must be done in a more sophisticated way to use models which allow you to calculate the real exposure to the liquid phase and to the cells, what you are giving to the patient later. Okay, then the next question, toxicological or biocompatibility assessment, you can call it as you like it. Here we are running in a quite interesting situation in the gene and cell therapy area. When a toxicologist today assesses impurities, and I talk about any impurities here, not only about pearls and things like that, then he has commonly data available in the literature from various cell-based test systems. So these are, you know, that Ames test and chromosome aberration tests, micronucleus tests, and you name it. Quite a range of test systems. In addition, he has commonly data from various test animals. That means feeding studies from rats and dogs, and sometimes also gavage and injection and things like that, with different endpoints, by the way. So different endpoints. And he has in silico data. And with that, our toxicologists are able to make quite reasonable risk assessments for impurities and are able to calculate PDE values and TDE values. Also, this is quite, I would say, a heterogeneous data set. They have quite good methodologies established to do that. So they can develop PDE values and TDE values based on this data. This changes quite significantly if you think about the gene and cell therapy area, because today the toxicologists have no methodology established which allow them to transfer, for example, Ames tests to isolated human or non-human cells which are manipulated ex vivo. This just doesn't exist. And the other thing is also there exists no data where you can say, okay, I have a rat study, and with that rat study I'm now evaluating what's going on in a T cell application. So this is something which doesn't exist. And the other thing is, of course, in a gene and cell therapy application, you do not only want to have no detrimental effects to your cells. You also do not want to have a stimulating effect in your cells. So in principle, the system shall be inert in such an application. And also for this, toxicologists have not really tools to investigate that. What do we know about interaction of pearls with cells? We know a little bit about that, and this is not so nice because we saw that there are some pearls which show effects on cells. For example, you know that famous bis-ditertzbutylphenol phosphate, which was detrimental to CHO cells. And we have also shown in an investigation published by my colleague Dana Budde that also T cells are sensitive to bis-ditertzbutylphenol phosphate. So also T cells are influenced by that. And we know also that pearls are adsorbed on cells. And as I said, this PhD student from me did some investigations on the adsorption of pearls on cells. And by the way, you can see nicely here, the bis-ditertzbutylphenol is that guy. Is also a guy which is very well adsorbing on the surfaces on cells. So we know that there are some troublemakers and we need to investigate that. For that purpose, we collaborated with the Max Planck Institute in Dortmund. They are using a high throughput cell painting assay, which goes back to a test from N. Carpenter from the MIT. This is a morphological test system where you incubate human cells, human osteosarcoma cells, with the impurities, and then you, or with actives, no? And then you stain that cells after a certain time, and then you make some morphological studies of the cells and subcellular environments inside the cells, and then you have a big computer system behind which is able to make a profiling of this information. To say, okay, certain changes in the morphology of the cells or subcellular particles cause this effect and that effect and so on. And the cool thing is, with such a test, you can immediately, with one study, investigate more than 1,000 cell features. So we are not relying here on one cell feature saying, okay, the cells are dying or the viability goes down or something like that. No. With one run, more than 1,000 cell features. And the method, and the Max Planck method here in Dortmund, they are able to trace from these 1,000 cell features, they are able to correlate that with effects, with more than 570 effects on cells, which can be caused if you imply impurities or actives. We can also use it for actives, of course, to the cells. So this test has a big advantage. It uses human cells. So what we really want to know, we want to know what's going on with ex vivo manipulated human cells. These morphological observations are associated with detrimental and stimulating effects. So we can study both in these tests. The endpoints are far away from cell tests, and we can also determine EC50 values if possible. The other things are we only need a small number of cells and low amounts of extracts are required. And the test is fast, is reliable, or everything is published and so on. And this is, in principle, is a test which is used in active screening in a quite appropriate way already. So, what did we exactly? We then used 45 prominent extractables. So from our database, it's very easy to say what are the most common extractables you find in the world. And then we saw there are 45 most prominent extractables, which are also water soluble, which is necessary in this case here, which has a certain water solubility. Okay. And we had 2 filter extracts from a Sartorius filter, a Sadopore filter, and a tubing. They were also applied to the test, to this cell painting test at the Max Planck Institute. In parallel, we run our standardized cell compatibility test. This is the Decima test with an industrial cell line. This was just for comparison. And then we got the results. And these results were, to be honest, quite a surprise for me in the beginning, but later I understood it a little bit better what's going on here. 40 out of these 45 compounds did not show any effect on these 1,000 cell features or these more than 500 effects we were studying. So the cells were completely unimpressed by our nice extractables. This was a good finding. We found one new compound which has indeed a very strong effect to these cells. I will show you soon. And we found 4 compounds which were more or less known to be detrimental to cells. And again on the next slide. And the results are more or less in line with the ASTM test, at least for the 4 compounds where we know already that they are critical. This is a list of our 45 nice compounds, and the people in the room here all familiar with extractable leachables know them very well. So there is our nice bis-tazbutylphenol phosphate, of course. There are tazbutanol. There are, I don't know, you know them. And in addition to extracts from a filter cartridge, from a polysulfone filter, and a 50% ethanol extract, so an USP extract, so to say, from a silicone tubing. And these are now the compounds, the only compounds highlighted which showed an effect. First of all, the most critical compound in this test was the lauryl D-ethanolamine. It shows a strong effect on the cholesterol homeostasis. As I said, we can trace back even to the effect. No? So it's possible to say what effect is caused. And it is lysosomotrophic, so it bursts the cells in the end. And we could determine an EC50 value for that compound. So for the other compounds, the effect was much weaker. That means we were not able to determine an EC50 value. So we saw an induction in the test, but it was quite low. Not low, it was there, but far from causing an effect on the EC50 value. So this is, of course, TPO. Some of you may know them. This is a compound which is used as photoinitiator for various compounds, for example, for tooth implants and things like that. Also single-use devices. Then we have our nice bis-detatsbutyl phosphate here on the list. We have 4-nonylphenol, also a compound which is already known to be not that nice, and again with the effect. And then caprolactam. This was a little bit a compound which was a little bit tricky because there was a test where we saw effects, but after repetition of the test, we could not reproduce that, and the induction was very, very low. It was only 55%, and 50% is the threshold. So this is more or less an outlier which needs to be skipped from that list here. So last but not least, I would say yes, it is also possible to establish reasonable tox tests and reasonable tox evaluations for gene and cell therapy applications, but we need, of course, new data for that. We need data from tests like that here, where we really expose human cells to these impurities and investigate what's going on with that impurities. And yeah, this is something what's needed. And then we can evaluate patient safety and process performance from that point of view. To come to a summary, I would say yes, it is possible to use extractables data as they are available today. They are comprehensive, they are scalable, they can be extrapolated, the solvent and the temperature are okay for our applications. Exposure calculations, as I said, these need a little bit more brain work behind to understand what's really going on in the liquid phase and cellular phase, in particular considering flow through the system and the growth of the biological material. And what you can also say, it is today possible, and these tests are available, to study effects on human cells with modern tests, and this shows that quite a range of PERLs, besides some troublemakers, have no effect on human cells. So all in all, and this is good for us as single-use device supplier, we can say, okay, we are quite confident that our devices are suitable to be used in the gene and cell therapy area. They allow a robust and safe application. And as we know, in principle, they are already the preferred devices to be used in gene and cell therapy. You can make a cross on that. And if you are interested in this work and would like to drill a little bit deeper into this work, this is already published. So there is a publication in the Pharma International on the giving approximately what I've talked about here, and this cell painting assay. This is published in Nature Scientific Report, where we published all these results. What I've shown here. So I was quite proud to see that Nature published this paper. So okay, and not only I was proud, also my colleagues were proud. So Dana Budde, Ina Pahl, and Roberto Menzel. Dana is our biocompatibility specialist. Ina and Roberto are the colleagues from the extractable leachables laboratory. I have, of course, to thank the Max Planck Institute, Dr. Anja Sievers and Dr. Axel Pahl for their collaboration. This was very fruitful. And also our toxicologist, Mr. Fruit, who commonly works with us and helped a lot in discussion of these results, what we have found here, and how we can apply them. Yeah, and with that, I'm at the end of my talk. Thanks.